So here's something kind of wonderful to think about as you're lying there, getting comfortable, letting the day fall away. Right now, as you settle in and your breathing starts to slow, your brain is quietly preparing to do something it's done every single night of your life, something it did last night and the night before that, stretching all the way back to the very first night you were born. And yet, for most of human history, nobody had the faintest idea it was happening. Nobody even suspected. You're about to enter a state so strange, so biologically peculiar, that when scientists finally stumbled onto it in the early 1950s, they didn't believe their own instruments. They thought the machines were broken. I'm talking about REM sleep, rapid eye movement sleep, and I want to walk you through everything about it tonight, how it was discovered, what's actually happening inside your brain when you're in it, why your body essentially paralyzes itself on purpose, what it has to do with dreaming, with memory, with learning, with emotion, with creativity, with survival itself. There's a lot here, and none of it is as simple as you might think. But that's okay. We've got all the time in the world. And honestly, the story of how we figured all this out, the people involved, the accidents, the arguments, the machines humming in dark laboratories at 3 a.m, that story alone is worth the telling. Let's start at the beginning. And by the beginning, I mean the time before anyone knew there were different kinds of sleep at all. For most of recorded history, sleep was treated as a single thing. You were either awake or you were asleep. That was it. 2 states, like a light switch. Aristotle thought sleep was caused by warm vapors rising from the stomach after eating, which, is kind of charming in its wrongness. The idea was that digestion produced these fumes. They rose to the brain, cooled down, and that cooling process made you drowsy. He actually wrote a whole treatise about it, called On Sleep and Sleeplessness, around 350 BC. And to be fair to Aristotle, he was doing his best without any of the tools we take for granted. He couldn't measure electrical activity in the brain. He couldn't peer inside a living skull. He was working from observation and logic, and he noticed that people get sleepy after meals, so he built a theory around it. Wrong theory, but a reasonable guess for the 4th century BC. For the next 2,000 years, not much changed. Sleep was still treated as this uniform, passive state. The brain was thought to basically shut down, like a factory closing for the night. Lights off, nobody home. Even into the early 20th century, most scientists assumed sleep was just the absence of wakefulness, that the brain was doing nothing interesting, just resting, recharging, waiting for morning. And then along came a machine that changed everything. In 1929, a German psychiatrist named Hans Berger published a paper that would eventually reshape our understanding of the brain, though at the time, almost nobody paid attention. Berger had spent years, literally years, starting around 1924, trying to record the electrical activity of the human brain using electrodes placed on the scalp. He called his invention the electroencephalogram, which we now just call the EEG, the electroencephalogram. And what Berger found was that the brain produces continuous, rhythmic electrical patterns. Even when a person is sitting still, doing nothing, eyes closed, the brain is humming with activity. He identified what he called alpha waves, these smooth, regular oscillations that appear when you're relaxed and awake with your eyes shut. And he noticed that when you opened your eyes or started concentrating on something, those waves changed. They got faster, less regular. Now, here's the thing about Berger. He was not a popular figure in the scientific community. He was secretive, a bit of a loner, and he had some, let's say, unconventional interests. He'd originally gotten into brain research because he believed in telepathy. He thought that if the brain produced electrical signals, maybe those signals could be transmitted between people. So when he published his EEG findings, a lot of his colleagues were skeptical. Some thought the electrical signals he was picking up were just artifacts, noise from the muscles in the scalp, interference from the equipment, that sort of thing. It took several years and independent replication by other researchers, particularly the British physiologist Edgar Adrian in 1934, before the scientific world accepted that yes, the brain really does produce measurable electrical waves, and yes, Berger's recordings were legitimate. But even after the EEG was accepted as a real tool, it took a while for anyone to think about using it to study sleep in a systematic way. The technology was cumbersome. The recording equipment was enormous. You had to glue electrodes to someone's head, hook them up to amplifiers and ink-writing oscillographs, these machines that trace the brain's electrical signals onto long, continuous rolls of paper. The paper scrolled out slowly, inch by inch, and the pens wiggled up and down, leaving these wavy lines. To record a full night of sleep, you needed hundreds of feet of paper. Literally. Someone had to sit there all night, monitoring the equipment, changing the paper rolls, making sure the pens didn't run dry or the electrodes didn't come loose. It was tedious, expensive, and most researchers didn't see the point. Sleep was boring, remember? The brain was just resting. Why waste a night watching someone do nothing? But a few researchers did start looking at sleep with the EEG, and what they found was immediately interesting. In the late 1930s, a scientist named Alfred Loomis, and Loomis is a fascinating character, by the way, a wealthy Wall Street financier who was also a serious amateur scientist with his own private laboratory in Tuxedo Park, New York. Loomis and his colleagues published a series of papers showing that sleep wasn't uniform at all. The brain's electrical patterns changed dramatically throughout the night. They identified several distinct stages, which they labeled A through E, based on the characteristics of the brainwaves. Stage A was drowsiness, where the alpha waves started to break up. Stage B was light sleep, with these slower, irregular patterns. Stages C and D showed progressively larger, slower waves, what we now call slow wave sleep or deep sleep. And stage E was the deepest, with these big, rolling delta waves, slow and powerful. This was a genuine revelation. Sleep wasn't one thing. It was multiple things, multiple states, and the brain cycled through them in patterns throughout the night. But even Loomis missed the biggest discovery. Even with all his equipment and all those rolls of paper, he missed what was happening during certain periods of the night when the brain seemed to wake back up. And this is where we get to the real story. The discovery of REM sleep. And it starts with a graduate student who was given what seemed like a throwaway project. The year is 1951. The place is the University of Chicago. A physiology professor named Nathaniel Kleitman. And Kleitman is one of those figures who deserves to be much more famous than he is, had been studying sleep for decades. He was arguably the world's leading sleep researcher, one of the only people who took sleep seriously as a scientific subject. He'd even once spent 32 days living in Mammoth Cave in Kentucky, deep underground with no natural light, trying to understand the body's internal clock. That was back in 1938. He and a research assistant lived in the cave, trying to shift their sleep-wake cycle to a 28-hour day instead of 24, to see if the body would adapt. It mostly didn't, which told him something important about how deeply wired our circadian rhythms really are. So by 1951, Kleitman was well established, and he had a graduate student named Eugene Oserinsky. Oserinsky was in his early 30s, a bit older than the typical grad student, and he'd had a winding path to get there. He'd served in the Army during World War II, tried dental school, dropped out, and eventually ended up in Kleitman's lab looking for a dissertation project. And Kleitman gave him one that nobody else wanted. He told Oserinsky to study the slow, rolling eye movements that happen as people fall asleep. These were well known. When you're drifting off, your eyes tend to make these lazy, wandering movements under your closed lids. Kleitman wanted someone to document them more carefully, maybe correlate them with the EEG patterns. It was considered a minor project. Routine, almost busy work. Oserinsky started by studying infants because babies sleep a lot and are relatively easy to observe. He'd watch their closed eyelids for signs of eye movement and note the timing, and he began to notice something odd. At certain points during sleep, the baby's eyes weren't making those slow, drifting movements at all. Instead, their eyes were darting around rapidly under their closed lids. Quick, jerky, side-to-side movements, almost like the babies were watching something, and these episodes seemed to come and go in a regular pattern. This was curious enough that Oserinsky decided to look for the same thing in adults. He started running overnight sleep studies, hooking up subjects with EEG electrodes on the scalp and additional electrodes near the eyes to track eye movements, a technique called electrooculography. And sure enough, he found the same thing. Several times during the night, his adult subjects would enter periods where their eyes moved rapidly back and forth. And when he looked at the EEG during these episodes, he got a shock. The brainwave patterns didn't look like sleep at all. They looked like wakefulness. The tracings showed fast, low-voltage irregular activity, the kind of pattern you'd expect from someone who was alert and paying attention to something. But these people were clearly asleep. Their eyes were closed. They weren't responding to the environment. They were lying still in bed. Oserinsky's first reaction was that something was wrong with the equipment. This is important because it tells you how unexpected the finding was. He didn't think, "Oh, I've discovered something new." He thought, "The machine is broken." He spent weeks checking the electronics, replacing components, recalibrating. The old equipment in Kleitman's lab was finicky. It was a surplus polygraph machine, not exactly state-of-the-art. But every time he fixed something and ran the study again, the same pattern appeared. Periods of rapid eye movement accompanied by brain activity that looked like waking. And then the episode would end. The eyes would go still, and the brain would settle back into the slower patterns of deeper sleep. And then, an hour or so later, it would happen again. When Oserinsky brought these findings to Kleitman, his advisor was skeptical. Kleitman had been studying sleep for over 20 years, and he'd never noticed anything like this. But to his credit, Kleitman didn't dismiss it. He told Oserinsky to keep going, to be more rigorous, to wake people up during these episodes and ask them what was happening. And that's when the second piece of the puzzle fell into place. When subjects were woken during these rapid eye movement periods, they almost always reported vivid dreams. Not vague, fragmentary impressions, but rich, detailed, story-like dreams. When they were woken during other stages of sleep, they usually reported nothing, or only the faintest wisps of thought. Think about what that meant. For the first time, there was an objective, measurable, physiological marker that correlated with dreaming. You could look at the squiggly lines on a piece of paper and say, with reasonable confidence, "This person is dreaming right now." Before this, dreams had been entirely subjective. You could only know about them if someone told you after the fact. Now there was a window into it, a way to watch dreaming happen from the outside. Oserinsky and Kleitman published their findings in 1953 in a short paper in the journal Science. It was only about 2 pages long. The title was dry and technical. Regularly occurring periods of eye motility and concomitant phenomena during sleep. Not exactly a grabby headline, but the implications were enormous. They had discovered an entirely new state of consciousness, one that the brain entered multiple times every single night, one that had been happening in every sleeping human being throughout all of history, and nobody had ever documented it before. And here's what's really fascinating about what happened next. You might expect that a discovery this significant would immediately set the scientific world on fire. But it didn't. Not right away. The paper got some attention, sure, but sleep research was still a backwater. Most physiologists were focused on other things. It would take another researcher, a young man named William Dement, who arrived in Kleitman's lab as a medical student right around the time Oserinsky was finishing up, to really push the discovery forward and begin mapping out what REM sleep actually was, how it fit into the architecture of a full night's sleep, and why it mattered so profoundly. Dement would go on to become perhaps the most important sleep researcher of the 20th century, and his work over the next decade would reveal things about REM sleep that were stranger and more important than anyone in that cramped Chicago laboratory could have imagined. And Dement, he was exactly the right person at the right time. He was a medical student, young, full of energy, and he had this quality that you sometimes see in people who end up changing a field. He was absolutely captivated by what he was seeing, not cautiously interested, not academically curious. Captivated. He later said that when he first watched a sleeping subject's eyes start darting back and forth beneath their eyelids, and then looked at the EEG trace showing this wild, active brain, he felt like he was witnessing something that should have been impossible. Here was a person lying completely still, seemingly dead to the world, and inside their skull, a storm was raging. Dement threw himself into the work, and what he did over the next few years was painstaking, methodical, and genuinely revelatory. He started running all-night sleep recordings, not just checking in on subjects periodically, but monitoring them continuously from the moment they fell asleep until the moment they woke up. Hour after hour, all through the night, watching the EEG paper scroll by. And what emerged from those long, bleary-eyed nights in the lab was the first real map of sleep architecture, the structure of how a whole night of sleep actually unfolds. Here's what Dement found, and this is something worth sitting with for a moment, because it's one of those things that seems simple once you hear it, but was completely unknown before someone actually did the work. Sleep isn't just a smooth descent into unconsciousness followed by a smooth return. It's cyclical. Your brain moves through distinct stages in a repeating pattern, and each cycle takes roughly 90 minutes. You fall asleep, and you descend through progressively deeper stages, what we now call stage 1, stage 2, stage 3, and sometimes stage 4, though modern classification has collapsed those last two together. Stage 1 is that drowsy threshold where you're barely under. Stage 2 is a lighter sleep, but clearly sleep, marked by specific EEG signatures called sleep spindles and K complexes, these little bursts and sharp waves that your brain produces. And then stage 3, or what's often called slow wave sleep, is the deep stuff. That's where your EEG shows these big, rolling, slow delta waves. Your brain is firing in these large, synchronized patterns, almost like the whole cortex is pulsing together. If someone tries to wake you during deep, slow-wave sleep, you'll be groggy and confused. It's hard to pull someone out of that state. But here's the thing. After you've been down in that deep sleep for a while, you don't just stay there. Your brain starts climbing back up through the lighter stages. And then, instead of waking up, something completely different happens. You enter REM. Your EEG suddenly looks like you're awake, fast, desynchronized, active. Your eyes start moving. And you dream. Dement documented this cycle happening over and over throughout the night, roughly every 90 minutes, give or take. But, and this is a detail that turns out to be really important, the cycles aren't identical. Early in the night, you spend more time in deep slow-wave sleep, and your REM periods are short, maybe 10 minutes or so. As the night goes on, the balance shifts. Your deep sleep gets shallower and shorter, and your REM periods get longer and more intense. By the last couple of cycles before you wake up in the morning, you might be spending 30, 40, even 45 minutes in REM at a stretch. That's why you're more likely to remember a dream if you wake up naturally in the morning. You're probably waking up right out of a long REM period. Think about what that means for a second. Your brain has this built-in program, this nightly schedule that it runs through with remarkable consistency. And it's not random. The architecture is structured, deliberate, as if different parts of the night are serving different functions. The deep, slow-wave sleep comes first, like the brain is prioritizing something. And we now think that's largely about physical restoration, growth hormone release, immune function, the clearing of metabolic waste from brain tissue. And then REM comes in waves that grow longer as the night progresses, as if the brain is saying, okay, we've taken care of the maintenance, now let's get to the other work, the dreaming work, the memory work, the emotional work. But we'll get to all of that. Dement published his findings on sleep cycles in 1957, and this time, people started paying more attention. He went on to found the first sleep disorders clinic at Stanford University in 1970, and he spent decades evangelizing for the importance of sleep research. He was tireless about it. He taught a famous course at Stanford on sleep and dreams that became one of the most popular classes on campus. He used to say that the most important thing he'd learned in his career was that sleep deprivation was a public health crisis that nobody was taking seriously enough. But that's a story for later. Let's stay with REM for now, because Dement's mapping of sleep architecture was just the beginning. Once researchers knew when REM happened and how often, the next question was obvious. What's actually going on during REM? What is the brain doing, and why? And this is where things start getting genuinely strange, because REM sleep, it turns out, is not just dreaming. It's a whole constellation of bizarre physiological events happening simultaneously, and some of them seem almost contradictory. Let's start with the one that's maybe the most dramatic. During REM sleep, your body is essentially paralyzed, not your diaphragm. You keep breathing, obviously, and not your eye muscles. Those are the ones doing all the rapid movements. But virtually every other voluntary muscle in your body goes limp. The technical term is atonia. Your brain actively sends signals that inhibit your motor neurons, and the result is that you are, for all practical purposes, unable to move. Now, why would your brain do that? Think about it. You're in a state where your brain is generating vivid, immersive experiences, dreams where you're running, fighting, flying, having conversations. If your muscles were active during all of that, you'd be acting out your dreams. You'd be thrashing around, kicking, swinging your arms, maybe getting out of bed and walking into walls. The paralysis is a safety mechanism. Your brain is essentially disconnecting itself from your body so it can run these intense internal simulations without any of them leaking out into actual physical movement. And we know this because of what happens when the mechanism breaks down. There's a condition called REM, sleep behavior disorder, where the atonia fails. The brain doesn't properly shut down the motor neurons during REM. And people with this condition do exactly what you'd predict. They act out their dreams. They punch, kick, shout, leap out of bed. It can be dangerous, both for the person and for anyone sleeping next to them. It was first formally described by Carlos Schenk and Mark Mahowald at the University of Minnesota in 1986, and studying it gave researchers a powerful confirmation that the paralysis of REM sleep isn't some incidental side effect. It's a critical feature. It's there for a reason. Here's what's also wild about the paralysis. Michel Jouvet, a French neuroscientist working in Lyon in the late 1950s and early 1960s, actually figured out where in the brain the paralysis signal comes from. Jouvet was doing experiments with cats, and I should mention that cats, like all mammals, have REM sleep too, which itself was a significant finding. Jouvet discovered that if he made very precise lesions in a specific part of the brainstem, a region called the pons, cats would enter REM sleep, but without the atonia. And what happened was remarkable and a little unsettling. The cats would be clearly asleep. Their EEG showed REM patterns. But they'd stand up, arch their backs, hiss at invisible threats, stalk around, even appear to pounce on imaginary prey. They were dreaming, and without the paralysis, their dream behavior was playing out in real time. Jouvet called this state paradoxical sleep, and that name actually stuck in French-language research for decades. He chose the word paradoxical because the whole thing seemed like a contradiction. A brain that looked awake inside a body that was supposed to be asleep. It was a paradox. Jouvet's work opened up a whole new line of investigation into the brainstem mechanisms controlling REM. It turned out that REM sleep isn't generated by the cortex, the thinking, conscious part of your brain. It's driven from below, from ancient structures deep in the brainstem, particularly the pons and areas around it. There are specific clusters of neurons that act almost like switches, turning REM on and off. Some of these neurons use the neurotransmitter acetylcholine to trigger REM, while others use norepinephrine and serotonin to suppress it. The interplay between these chemical systems creates the cycling you see throughout the night. REM turns on, runs for a while, gets shut off, and then the non-REM stages take over until the balance shifts again and the next REM period begins. And this is a really important point, so let me say it another way. REM sleep is not something your cortex decides to do. It's not a higher-level cognitive choice. It's driven by some of the oldest, most evolutionarily ancient parts of your brain. The brainstem is the part we share with reptiles, with birds, with every vertebrate that has ever lived. The machinery for REM is deep and old, and that tells you something about how fundamental it is. This isn't some luxury feature that evolution tacked on recently. This is core infrastructure. Now, while all of this brainstem switching is happening, while your body is paralyzed and your eyes are darting around, something else is going on that's equally fascinating. Your autonomic nervous system, the part that controls things you don't consciously manage, like heart rate, breathing, blood pressure, goes a little haywire during REM. Your heart rate becomes irregular. Your breathing gets uneven, sometimes faster, sometimes slower, with odd pauses. Your blood pressure fluctuates. Your body's ability to regulate its own temperature actually diminishes during REM. you become slightly more like a cold-blooded animal, less able to thermoregulate. If the room is too hot or too cold, your body is less equipped to compensate during REM than during other sleep stages. It's as if, in order to run these intense dream simulations, your brain has to temporarily relax its grip on some of the basic housekeeping functions it normally manages so tightly. And then there's what's happening in the brain itself, beyond the EEG patterns. Modern neuroimaging, PET scans, fMRI, has shown that during REM sleep, certain parts of the brain are actually more active than they are when you're awake. The amygdala, which is deeply involved in processing emotions, especially fear and anxiety, lights up intensely during REM. The visual cortex is highly active, which makes sense given how visually vivid dreams tend to be. But the prefrontal cortex, the part of your brain responsible for logical thinking, planning, self-awareness, rational judgment, that goes relatively quiet. And that explains so much about the nature of dreams, doesn't it? You're in this state where emotions are running high, visual imagery is vivid and immersive, but the rational, critical thinking part of your brain is essentially offline. That's why dreams feel so real while you're in them. That's why you can be flying through the air or talking to someone who died years ago, and it doesn't strike you as strange. The part of your brain that would normally say, "Wait, this doesn't make sense," is taking a break. So you've got this extraordinary state, a brain that's electrically active, emotionally charged, visually creative, but logically uncritical. A body that's completely paralyzed except for the eyes and the diaphragm, autonomic systems running loose, and all of this happening on a predictable schedule, multiple times a night, with the periods getting longer and more intense toward morning. It's one of the most complex physiological states that exists, and it happens to you every single night. But here's the question that haunted researchers for decades after Oserinsky and Kleitman and Dement and Jouvet laid down the basic description. Why? What is REM sleep actually for? Why would evolution build something this elaborate, this metabolically expensive, this physiologically dramatic, and run it every single night in every mammal and bird that's ever been studied? There had to be a function, maybe several functions, and figuring that out required a completely different kind of experiment, one that involved taking REM sleep away and seeing what happened when you took it away from someone. And that's exactly what researchers started doing in the late 1950s and into the 1960s. The method was straightforward, if a little cruel. You bring someone into the sleep lab, you wire them up with electrodes so you can monitor their brain waves in real time, and every time they slip into REM sleep, every time you see those fast, desynchronized waves and those darting eye movements, you wake them up. You let them fall back asleep. You let them cycle through the lighter stages. You let them sink into deep, slow-wave sleep. But the moment REM begins, you pull them out again. You're not depriving them of sleep entirely. You're surgically removing just this one specific phase and leaving everything else more or less intact. And what you're really asking is, does the brain need this? What breaks when you take it away? William Dement himself was one of the first to run these experiments systematically, starting around 1960. And the results were striking, though not always in the ways people expected. The most immediate and consistent finding was something called REM rebound. When you deprive someone of REM sleep for several nights, and then finally let them sleep undisturbed, they don't just return to their normal amount of REM, they gorge on it. The brain dramatically increases both the frequency and the duration of REM periods, as if it's trying to make up for lost time. On a recovery night, someone might spend 40 or even 50% of their sleep in REM, compared to the usual 20 to 25%, and the brain doesn't just passively drift into REM more often. It actively fights to get there. During the deprivation period itself, the attempts to enter REM become more and more frequent. On the first night, you might need to wake someone 5 or 6 times. By the third or fourth night, you might be waking them 20, 30, even 40 times. The pressure builds. The brain is insisting on this state with increasing urgency, like a spring being compressed tighter and tighter. Think about what that tells you. If REM sleep were just some quirky byproduct of brain activity, some evolutionary leftover with no real function, you wouldn't expect the brain to fight so hard to get it back. The existence of REM rebound is itself a powerful argument that REM serves a vital purpose. The brain keeps a running tally of how much REM it's gotten, and when it falls behind, it compensates aggressively. That's not the behavior of a system that's optional. Now, the behavioral effects of REM deprivation in those early human studies were real but subtler than people sometimes claim. Dement reported increased irritability, difficulty concentrating, and in some cases mild anxiety or emotional instability. There were sensational newspaper stories at the time suggesting that REM deprivation could drive people insane, and that's an exaggeration. The early studies didn't show psychosis or anything close to it in healthy subjects over the relatively short deprivation periods they used, which were typically 3 to 5 nights. But the emotional effects were consistent enough to notice. People became edgier, more reactive, more emotionally volatile. Their ability to regulate their feelings seemed to fray around the edges. The animal studies, though, those told a darker story. In the 1980s, Alan Rechtschaffen at the University of Chicago, working in the same institution where Kleitman had made his discoveries decades earlier, developed an ingenious, if unsettling, method for depriving rats of sleep. He used a rotating disc suspended over water. 2 rats lived on the disc, an experimental rat and a control rat. When the experimental rat's brain waves showed it entering REM, the disc would rotate, forcing both rats to walk to avoid falling into the water. The control rat was disturbed too, but it could sleep during the periods when the experimental rat was awake, so it accumulated significantly more sleep overall. The experimental rats, deprived of REM and eventually of all sleep, deteriorated badly. They lost weight despite eating more. Their fur became matted and patchy. They developed skin lesions. Their body temperature regulation fell apart. And eventually, after about 2 to 3 weeks, they died. Every single one of them. Now, it's important to be honest about what those experiments actually showed. The rats were deprived of all sleep eventually, not just REM, so you can't cleanly attribute everything to REM loss alone. And the stress of the experimental setup itself was significant. But the studies did demonstrate that sleep deprivation, including REM deprivation, was not merely uncomfortable. It was, in extreme cases, fatal. The body simply could not sustain itself without this state. Something essential was happening during sleep that kept the organism alive, and removing it had catastrophic consequences. So researchers knew REM mattered. The brain demanded it. The body suffered without it. But what exactly was it doing? What function was so important that evolution would build this elaborate, energy-intensive, neurologically dramatic state and run it every single night? This is where the story branches into several parallel lines of investigation, because it turns out REM sleep probably doesn't do just one thing. It does several things, and different research groups spent decades chasing different pieces of the puzzle. Let's start with memory, because that's one of the richest and most extensively studied areas. The idea that sleep helps with memory isn't new. People have suspected it for centuries. But the modern scientific investigation really picks up in the 1990s and 2000s when researchers started designing clever experiments to tease apart exactly which types of memory benefit from which stages of sleep. Here's the thing you need to understand about memory. It's not one monolithic process. There are different kinds of memory, and they seem to depend on different brain systems. You've got declarative memory, facts and events, things you can consciously recall, like the capital of France or what you had for dinner last Tuesday. And you've got procedural memory, skills and habits, things your body knows how to do, like riding a bike or playing a piano scale. And within declarative memory, there's a further distinction between episodic memory, which is your personal autobiography, and semantic memory, which is general knowledge about the world. What researchers found, and this took years of careful work by groups including those led by Robert Stickgold at Harvard and Jan Born in Germany, is that different sleep stages seem to preferentially support different types of memory. Deep, slow-wave sleep appears to be particularly important for consolidating declarative memories, facts, events, the stuff you studied for an exam. But REM sleep seems especially critical for procedural learning, for emotional memory processing, and for something even more interesting, the integration and creative recombination of memories. Stickgold's work is particularly elegant here. In one line of experiments, subjects were trained on a visual texture discrimination task. Basically, they had to spot subtle patterns on a screen. Their performance was tested, then they slept, and then they were tested again. And here's what was remarkable. Performance didn't just stay the same after sleep. It actually improved. People got better at the task overnight without any additional practice. And the amount of improvement correlated specifically with the amount of REM sleep they got, particularly late-night REM sleep. Those long, intense REM periods that happen in the last few hours before you wake up. If you deprived people of that late-morning REM by waking them up early, the improvement vanished. Think about what that means for a moment. Your brain isn't just passively storing what you learned during the day. During REM sleep, it's actively working on it. It's refining the neural circuits, strengthening the connections, essentially practicing the skill while you lie there motionless. You go to bed mediocre at something and wake up measurably better, and the improvement happens specifically during REM. But memory consolidation, as important as it is, might not even be the most fascinating thing REM sleep does with your memories. There's a growing body of evidence that REM sleep is when your brain does something you might call creative integration. During REM, the brain seems to take newly acquired information and test it against your existing knowledge, looking for connections, patterns, and associations that you might not have noticed while you were awake. It's as if your brain is running a kind of loose associative search through everything it knows, seeing what fits with what. This might explain why people so often report solving problems in their dreams or waking up with creative insights after a good night's sleep. There's a famous, possibly apocryphated but illustrative, story about the chemist Auguste Kekulé, who claimed he discovered the ring structure of benzene after dreaming of a snake seizing its own tail. Whether or not that specific anecdote is literally true, the broader phenomenon is well documented. Researchers have shown in controlled experiments that sleep, and REM sleep in particular, enhances the ability to find hidden rules in data sets, to make remote associations between seemingly unrelated concepts and to restructure problems in ways that reveal solutions. One beautiful study by Wagner and colleagues, published in Nature in 2004, trained subjects on a math task that had a hidden shortcut, a pattern in the numbers that, if you noticed it, would let you solve problems much faster. After training, some subjects slept for 8 hours and some stayed awake. Of those who slept, about 60% discovered the hidden shortcut when retested, compared to only about 25% of those who stayed awake. Sleep more than doubled the rate of insight, and the researchers found evidence linking this specifically to REM sleep. So here's what seems to be happening. During REM, your prefrontal cortex, remember, the logical, rule-following part, is quiet, but your associative networks are highly active. Your brain is free to make connections that your waking, rational mind might reject as too unlikely or too weird. It's trying out combinations, testing associations, playing with possibilities, in a way that the more disciplined waking brain wouldn't allow. And some of those wild combinations turn out to be genuinely useful. That's creativity. That's insight. And it might be one of the reasons REM sleep exists. But there's another function of REM sleep that might be even more fundamental to your daily well-being, and that's emotional processing. This is where the work of Matthew Walker at UC Berkeley, among others, has been particularly illuminating. Remember that during REM sleep, the amygdala, your brain's emotional alarm system, is highly active, and the prefrontal cortex, which normally modulates and controls emotional responses, is dampened. At first glance, that seems like a recipe for emotional chaos. But Walker and his colleagues proposed something quite elegant, that REM sleep serves as a kind of overnight therapy session. The idea goes like this. During REM sleep, your brain reactivates emotional memories from the day, the argument you had with a coworker, the embarrassing moment at lunch, the anxiety about an upcoming deadline. But it reactivates them in a neurochemical environment that's fundamentally different from waking life. Specifically, norepinephrine, the brain's version of adrenaline, the chemical that gives emotional experiences their visceral, heart-pounding, stomach-churning intensity, is almost completely absent during REM sleep. It's one of the only times in the entire 24-hour cycle when your brain is essentially norepinephrine-free. So you're replaying emotional memories, but without the chemical that makes them feel emotionally charged. And Walker's hypothesis is that this is precisely the point. By reprocessing emotional experiences in this calm neurochemical context, REM sleep gradually strips the sharp emotional edge off difficult memories. You still remember what happened. The memory itself is preserved, but the gut-wrenching feeling that went with it is diminished. You wake up and the problem is still there, but it doesn't sting quite as much. Time heals all wounds, people say. But maybe it's not time. Maybe it's REM sleep. There's experimental support for this. Walker's group showed that when people view emotionally disturbing images and then get a full night's sleep with plenty of REM, their emotional reactivity to those images decreases significantly the next day. But if you deprive them of REM sleep specifically, the emotional charge remains. The memories stay hot. And in brain imaging studies, you can see that after REM-rich sleep, the amygdala's response to those same emotional images is reduced, while the prefrontal cortex, the rational regulator, shows increased connectivity and control. It's as if REM sleep recalibrates the emotional circuits overnight, restoring a healthier balance between feeling and thinking. This has profound implications, and some of them are a little unsettling. Think about post-traumatic stress disorder, for instance. One of the hallmark symptoms of PTSD is that traumatic memories don't fade emotionally the way normal memories do. They stay raw, vivid, and viscerally distressing months or even years after the event. People with PTSD also tend to have severely disrupted REM sleep, fragmented, shortened, plagued by nightmares that replay the trauma with full emotional intensity. And this raises a haunting question. Is the disrupted REM sleep a symptom of PTSD, or is it part of the cause? Is the disorder persisting precisely because the brain can't complete its normal REM-based emotional processing? Walker and others have suggested that this might be exactly what's happening, that PTSD is, at least in part, a failure of the REM sleep emotional processing system. The overnight therapy session keeps getting interrupted, so the wound never heals. And if that's true, it opens up entirely new ways of thinking about treatment. Some researchers have started looking at whether enhancing REM sleep, or at least protecting it from disruption, might help people recover from trauma more effectively. The drug prazosin, which blocks norepinephrine receptors and was originally developed for high blood pressure, has shown promise in reducing trauma-related nightmares in PTSD patients. And the mechanism might be precisely that it helps restore the low norepinephrine environment that normal REM sleep provides, the environment the brain needs to do its emotional processing work. But here's where the story gets even more layered, because emotional processing and memory consolidation aren't really separate functions. They're deeply intertwined. The memories that REM sleep seems most interested in processing are emotional memories, and the creative associations that REM enables often involve emotionally significant material. So, rather than thinking of REM as having 3 or 4 distinct jobs, it might be more accurate to think of it as running a single, extraordinarily sophisticated process, one that simultaneously consolidates important experiences, extracts their emotional significance, integrates them with existing knowledge, and recalibrates your emotional responses. All at once, all while you lie there with your eyes darting beneath your lids and your muscles completely still. And there's yet another dimension to REM sleep's function that researchers have been exploring. One that takes us back to the very beginning of life. Not your life, but life on Earth. Because when you look at REM sleep across species, something curious emerges about when and how it evolved, and what it might tell us about why our brains need it so badly. Because REM sleep isn't unique to humans, not even close. But the pattern of who has it and who doesn't, that's where things get really interesting. Let's start with mammals, because that's the group we know best. Almost every mammal studied so far shows clear evidence of REM sleep. Dogs, cats, rats, elephants, whales, bats, opossums, they all have it. If you've ever watched a dog sleep, you've probably seen the twitching paws, the little muffled barks, the rapid eye movements flickering beneath closed lids. That's REM. Your dog is dreaming, or at least going through the same neurological process that produces dreams in humans. And here's the thing. The amount of REM sleep varies enormously across species, and the reasons behind that variation have puzzled researchers for decades. Consider the platypus. This strange, egg-laying mammal from Australia holds a kind of record in the sleep world. It spends an astonishing amount of time in what appears to be REM sleep, possibly up to 8 hours a day. That's far more than any other mammal we know of. Now, the platypus is a monotreme, one of the most ancient lineages of mammals still alive. It diverged from the rest of the mammalian family tree, something like 166 million years ago. and the fact that it has so much REM sleep has led some researchers to speculate that REM might be an ancient feature of mammalian brains, something that was present very early in mammalian evolution and has been conserved, modified, and fine-tuned ever since. But then you look at birds, and the picture gets more complicated in the best possible way. Birds also show REM sleep. Their eyes move, their muscle tone drops, though not as completely as in mammals, and their brain activity shifts into patterns that look remarkably similar to wakefulness. Some songbirds even appear to rehearse their songs during REM sleep. Researchers at the University of Chicago, and yes, it's that same University of Chicago that keeps showing up in sleep research, found that neurons in the brains of zebra finches fire during REM sleep in patterns that mirror the neural sequences the birds produce when they're actually singing. It's as if the bird is practicing its song in its sleep, running through the motor sequences without actually producing sound. Think about what that means for a second. That's essentially the same kind of procedural memory consolidation we talked about earlier with humans learning new motor skills, except it's happening in a bird brain that diverged from the mammalian lineage over 300 million years ago. And that's the puzzle. Birds and mammals share a common ancestor way, way back, a reptile-like creature that lived during the Carboniferous period. But the lineages split long before either group developed the complex brains they have today. So did REM sleep evolve once in that ancient common ancestor and get inherited by both birds and mammals? Or did it evolve independently, twice, because the underlying need it serves is so fundamental that evolution converged on the same solution in two completely separate lineages? Most researchers today lean toward the second explanation, convergent evolution. The thinking goes something like this. Both birds and mammals independently evolved warm-bloodedness, relatively large brains for their body size, and complex behaviors. And both groups, facing similar challenges of learning, memory consolidation, and neural maintenance, independently stumbled onto REM sleep as a solution. If that's true, it tells you something profound about REM. It suggests that once a brain reaches a certain level of complexity, REM sleep, or something very much like it, becomes not just useful, but necessary. Evolution invented it twice because the problem it solves is that important. Now, what about reptiles? This is where the story gets murkier, and honestly, more fascinating. For a long time, the conventional wisdom was that reptiles don't have REM sleep. Their sleep looked simpler, just a general quieting of brain activity without the dramatic cycling between stages that mammals show. But in 2016, a study by Gilles Laurent and his colleagues at the Max Planck Institute for Brain Research in Frankfurt changed the conversation. They recorded brain activity in Australian bearded dragons, those calm, slightly prehistoric-looking lizards you sometimes see in pet stores, and found something unexpected. The dragons showed two alternating states during sleep, one that resembled slow-wave sleep, and another that looked a lot like REM. The cycles were much shorter than in mammals, about 80 seconds each, compared to our 90-minute cycles. But the pattern was there. Eye movements, changes in brain activity, the whole signature. This finding was genuinely surprising, and it's still being debated. Some researchers argue that what Laurent's team found is a true homologue of mammalian REM, meaning it's the same basic process, inherited from a common ancestor after all, which would push the origin of REM sleep back to at least 320 million years ago, before reptiles, birds, and mammals diverged. Others argue that the dragon sleep states might be superficially similar but mechanistically different, Another case of convergent evolution, where the brain independently arrives at similar-looking solutions. The honest answer is, we don't know yet. But either way, the implication is remarkable. Either REM sleep is incredibly ancient, stretching back to the earliest land vertebrates, or it's so useful that evolution keeps reinventing it across wildly different lineages. Both possibilities point to the same conclusion. Whatever REM sleep does, it matters enormously. And here's where the evolutionary perspective connects back to all those functions we've been talking about. The memory consolidation, the emotional processing, the creative integration. Because when you think about what all those functions have in common, a pattern starts to emerge. They're all about making sense of experience, taking raw information that came in during the day, sorting through it, finding connections, stripping away what's irrelevant, strengthening what's important, and integrating it all into a coherent model of the world. That's not a luxury. For any animal with a complex brain navigating a complex environment, that's survival. Think about a young songbird learning its species' song. It has to listen to adult males singing, store those auditory patterns, and then gradually shape its own vocal output to match. That's a learning process that unfolds over weeks, and the evidence suggests that REM sleep is when the brain does much of the behind-the-scenes work of refining those motor programs. Or think about a rat exploring a new environment, mapping out where food is, where predators lurk, where the safe paths are. During subsequent REM sleep, the rat's hippocampus replays the neural patterns associated with that exploration, sometimes at altered speeds, sometimes in novel combinations, as if the brain is running simulations, testing what it learned, integrating the new spatial information with what it already knows. This idea, that REM sleep is fundamentally about simulation, has been developed into a formal theory by some researchers. The neuroscientist Antti Revonsuo at the University of Turku in Finland proposed what he calls the threat simulation theory of dreaming. His argument is that dreams, which occur most vividly during REM sleep, evolved primarily as a way for the brain to simulate threatening scenarios. By rehearsing dangerous situations in the safe environment of sleep, the brain could prepare more effective responses for real encounters. It's like a flight simulator for survival. And when you look at dream content across cultures and across species, to the extent we can infer anything about animal dreams from their neural activity, threat-related themes do seem disproportionately common. People dream about being chased, about falling, about social conflicts, about losing their way. These aren't random. They're the kinds of scenarios that, for most of human evolutionary history, could get you killed. Now, Revonsuo's theory is just one framework, and it doesn't explain everything about REM sleep. It doesn't fully account for the emotional processing dimension, or the creative insight dimension, or the many non-threatening dreams people have. But it captures something important about the evolutionary logic. REM sleep isn't idle time. It's the brain doing critical work that natural selection has deemed important enough to protect with homeostatic mechanisms, important enough to evolve independently in multiple lineages, important enough that depriving an animal of it triggers an increasingly desperate rebound response. And speaking of that rebound response, there's something else worth noting about how REM sleep is distributed across the lifespan, because this might be the most striking clue of all about what REM is really for. If you look at how much time different age groups spend in REM sleep, the pattern is dramatic. A human newborn spends roughly half of its total sleep time, about 8 hours a day, in REM sleep. A premature infant, born a few weeks early, spends even more. By age 5, REM sleep has dropped to about 2 hours a night. By adulthood, it's down to roughly 90 minutes to 2 hours. And in old age, it decreases further. Think about what that means. The period of life when the brain is developing most rapidly, when neural connections are being formed, pruned, and refined at an astonishing rate, is also the period when REM sleep is most abundant. This has led to what's sometimes called the ontogenetic hypothesis, proposed in the 1960s by Howard Rothward, Joseph Muzio, and William Dement. Their idea was that REM sleep provides a source of internal stimulation for the developing brain. During fetal development and early infancy, the brain needs activity to wire itself properly. Neurons that fire together wire together. That's a fundamental principle of neural development. But a fetus doesn't have much external stimulation. It's floating in the dark, in a warm, quiet environment. So where does the neural activity come from that drives brain development? Rothward and his colleagues proposed that REM sleep generates it internally. The bursts of neural activity during REM, the activation of the visual cortex, the motor cortex, the emotional centers, serve as a kind of self-generated stimulation that helps the brain build and test its own circuits. And there's evidence to support this. In studies with young animals, kittens for instance, depriving them of REM sleep during critical developmental windows leads to abnormalities in the visual cortex. The neural circuits don't develop properly without that internal stimulation. It's as if REM sleep is the brain's way of bootstrapping itself, running test patterns through its own hardware to make sure everything is wired correctly before the organism needs to rely on those circuits for real-world survival. This developmental role doesn't contradict the memory consolidation and emotional processing functions we talked about earlier. It complements them. In infancy, REM sleep is primarily about building the brain's basic architecture. As the brain matures and begins accumulating experiences, REM sleep gradually shifts its emphasis toward processing those experiences, consolidating memories, extracting patterns, calibrating emotional responses. The machinery is the same, but the job description evolves as the brain itself evolves. It's a beautifully elegant system when you step back and look at it. And it raises a question that researchers are still grappling with today, one that connects the evolutionary story to the cutting edge of neuroscience. If REM sleep is doing all of this critical work, building the brain in infancy, processing memories and emotions in adulthood, potentially simulating threats and rehearsing skills across the entire lifespan, then what exactly is happening at the level of individual neurons and synapses during those darting-eye, muscle-paralyzed, dream-filled minutes? What is the actual mechanism? Because describing what REM sleep does is one thing. Understanding how it does it, at the cellular and molecular level, is something else entirely. And that's a question that has driven some of the most remarkable neuroscience of the past 20 years, starting with a discovery about what happens to the connections between your neurons while you sleep. Because it turns out that the synapses in your brain, those tiny gaps between neurons where chemical signals jump from one cell to the next, don't just sit there passively waiting for instructions. They're dynamic. They grow stronger with use, they weaken with neglect, and during sleep, something truly fascinating happens to them that researchers didn't fully appreciate until relatively recently. The story starts with a concept called synaptic homeostasis, and the two people most associated with it are Giulio Tononi and Chiara Cirelli, both at the University of Wisconsin-Madison. They've been working on this idea since the early 2000s, and it's one of those hypotheses that sounds almost too simple at first, but gets more compelling the longer you sit with it. Here's the basic idea. Every day, while you're awake, your brain is learning things. Not just big, dramatic lessons, not just studying for an exam or memorizing a phone number. Every single sensory experience, every conversation, every moment of navigating your environment strengthens certain synaptic connections. That's what learning is, at the cellular level. Neurons that fire together wire together, as the old saying goes. And over the course of a full waking day, the net effect is that synapses across your brain get stronger and stronger. They get potentiated, to use the technical term. Now here's the problem. Strengthening synapses takes energy. Stronger synapses require more neurotransmitter, more receptor proteins, more cellular machinery to maintain. If your synapses just kept getting stronger day after day after day, the energy demands would become unsustainable. Your brain already uses about 20% of your body's energy, despite being only about 2% of your body weight. It can't afford to let synaptic strength just ratchet up indefinitely. Plus, if every synapse is screaming at maximum volume, you lose the ability to distinguish signal from noise. The whole point of having some connections stronger than others is that the pattern of relative strengths encodes information. If everything is equally loud, nothing stands out. It's like trying to read a page where every word is bolded and highlighted. The emphasis becomes meaningless. So Tononi and Cirelli proposed that sleep, and particularly deep, slow-wave sleep, though REM plays a role too, is when the brain does a kind of global renormalization. It scales everything back down. It weakens synapses across the board, proportionally, so that the ones that were strengthened the most during the day still stand out relative to the others, but the overall level comes back to a sustainable baseline. Think of it like adjusting the volume on a mixing board. If every channel has been slowly creeping up all day, you don't need to change the relative balance between the channels. You just need to bring the master fader down so the whole system is back in a workable range. They called this the synaptic homeostasis hypothesis, or SHY for short, and they published the core framework around 2003. And what's elegant about it is that it explains something that had puzzled sleep researchers for a long time. Why does sleep deprivation impair learning? If sleep were just about rest, just about giving your body a break, then why would your ability to form new memories suffer so dramatically when you don't sleep? The synaptic homeostasis hypothesis gives you a clean answer. If you haven't slept, your synapses are still saturated from yesterday's learning. They're already near their maximum strength. There's no room to encode new information because the system hasn't been reset. It's like trying to write on a whiteboard that hasn't been erased. The surface is full. Now, you might be thinking, okay, but we've been talking about REM sleep specifically, and this synaptic scaling down sounds like it's mainly a slow-wave sleep thing. And you're right to notice that. Tononi and Cirelli's original formulation did emphasize slow-wave sleep because the slow oscillations, those big, rolling waves of synchronized neural activity that characterize deep sleep, seemed like the perfect mechanism for globally weakening synapses. When a large population of neurons alternates between firing together and going silent together, that rhythmic pattern could systematically downscale synaptic strength. But here's where it gets interesting for our purposes. REM sleep appears to do something more selective. Rather than a global scaling down, REM may be involved in a more targeted process, strengthening certain connections while pruning others. And this connects back to everything we've been talking about: memory consolidation, emotional processing, creative integration. During REM sleep, the brain isn't doing a blanket reset. It's doing something more like editing. It's deciding which of the day's new synaptic changes are worth keeping and which can be let go. Some of the evidence for this comes from molecular studies. During REM sleep, there's increased expression of certain genes associated with synaptic plasticity, genes involved in building and maintaining the structural proteins that make synapses stronger. One family of these genes codes for what are called immediate early genes, and their expression patterns during REM sleep suggest that specific circuits are being selectively reinforced. It's not everything getting turned down. It's certain things getting locked in. And this dovetails beautifully with some work by Marcos Frank, who was at the University of Pennsylvania and later at Washington State University. Frank studied the visual cortex development in kittens, similar to those deprivation studies we touched on, and found that REM sleep was specifically required for a process called ocular dominance plasticity. This is the process by which the visual cortex learns to properly integrate signals from both eyes during early development. What Frank showed was that it wasn't just sleep in general that mattered. It was specifically REM sleep. During REM, the molecular signals that consolidate the day's visual learning were most active. Block REM sleep, and those molecular consolidation signals didn't fire properly, and the visual cortex didn't develop normally. Allow REM sleep to occur, and you could actually watch the molecular machinery of long-term synaptic change kick into gear. So you start to get this picture where slow-wave sleep and REM sleep are doing complementary but different things at the synaptic level. Slow-wave sleep is the broad reset, the global scaling down that keeps the system sustainable. REM sleep is the selective reinforcement, the careful editing that decides which new connections deserve to become permanent. And the two stages alternate throughout the night in those 90-minute cycles that Dement mapped out all those decades ago, which means your brain is essentially oscillating between these two modes: scale down, selectively strengthen, scale down, selectively strengthen. Each cycle refines the process a little further. There's another molecular player in this story that's worth knowing about, and it's one you might not expect: brain-derived neurotrophic factor, or BDNF. This is a protein that acts like fertilizer for neurons. It promotes the growth and survival of synapses, and it's crucial for learning and memory. During waking, when you're actively learning, BDNF levels increase in the brain regions involved in whatever you're learning. And during subsequent REM sleep, BDNF signaling appears to be involved in consolidating those changes, in making the temporary synaptic strengthening from the day into something more durable. A really compelling study on this came from Jian and colleagues, who showed that blocking BDNF signaling during REM sleep in rats impaired their ability to consolidate fear memories. The rats could learn the association between a tone and a mild shock just fine while awake. But if BDNF was blocked during the REM sleep that followed, the memory didn't stick. The learning happened, but the consolidation didn't, which tells you that REM sleep isn't just passively allowing consolidation to occur, it's actively driving it through specific molecular pathways. And then there's the role of something called the mTOR pathway, mechanistic target of rapamycin, if you want the full name, though nobody actually calls it that in conversation. This is a molecular signaling cascade that's involved in protein synthesis at synapses. When a synapse needs to be permanently strengthened, not just temporarily boosted, but physically remodeled with new proteins to make the change last. The mTOR pathway is one of the key mechanisms that makes that happen. And guess when mTOR signaling is particularly active? During REM sleep. Specifically, during the REM sleep that follows a learning experience. Tudor Bott and colleagues at NYU showed this in some elegant experiments where they could track mTOR activation in specific neurons. After mice learned a new motor task, the neurons involved in that task showed increased mTOR signaling during subsequent REM sleep. And if you disrupted REM sleep, or if you pharmacologically blocked the mTOR pathway, the motor memory didn't consolidate properly. The mice would wake up the next day, and it was as if they'd only partially learned the task, even though their performance had been fine before they went to sleep. So now you can start to see the mechanism coming into focus. It's not magic. It's not mysterious. It's biochemistry. During REM sleep, specific molecular pathways, BDNF, mTOR, immediate early genes, and others, are activated in the neurons and synapses that were most active during the day's learning. These pathways trigger protein synthesis, which physically remodels the synapses, making temporary changes permanent. Meanwhile, synapses that weren't tagged for preservation during the day's experiences get weakened during slow-wave sleep's global downscaling. The net result is that by morning, your brain has a cleaner, more efficient, more refined version of yesterday's learning encoded in its synaptic architecture. And this brings us to one of the most visually stunning pieces of evidence in all of sleep science, even though we obviously can't see it right now. But I want you to imagine it. In 2017, a team led by Graham Dyring in Tononi and Sorelli's lab published a study in Science where they actually measured the size of synapses in the brains of mice after periods of sleep versus wakefulness. They used electron microscopy to image thousands of individual synapses, measuring the area of the synaptic interface, the actual physical contact zone, where one neuron communicates with another. And what they found was striking. After sleep, synapses were on average about 18% smaller than after an equivalent period of wakefulness. Not all synapses, though. The largest synapses, the strongest connections, presumably the ones encoding the most important or well-established information, were largely spared. It was the small and medium synapses that shrank the most. The brain was being selective. It was preserving its most critical connections while trimming the noise. 18% might not sound like much, but when you multiply it across the roughly 100 trillion synapses in the human brain, the cumulative effect on energy consumption, on signal-to-noise ratio, on the brain's capacity to encode new information the next day, it's enormous. And this process is happening every single night. Every time you close your eyes and drift through those cycles of slow wave and REM sleep, your brain is physically reshaping itself at the nanometer scale. Now here's something that connects this molecular story back to the emotional processing we talked about earlier, and it involves a brain region that keeps showing up in REM sleep research like a recurring character in a novel: the amygdala. Remember, neuroimaging studies showed that the amygdala is highly active during REM sleep, more active in some studies than during waking emotional experiences. And the prefrontal cortex, which normally keeps the amygdala in check, is largely offline. So you have this situation where the brain's emotional center is firing intensely without its usual regulatory oversight, while the molecular machinery of synaptic consolidation is running full tilt. What does that combination produce? Well, one theory, and this is where the work of Matthew Walker and others comes back into the picture, is that this is precisely the setup needed for emotional memory processing. The amygdala reactivates the emotional memories from the day, replaying them in a neurochemical environment that's fundamentally different from waking. Remember, norepinephrine is essentially absent during REM sleep. So the emotional content of the memory is being reprocessed, and the synaptic connections encoding that memory are being edited by the molecular consolidation machinery, but without the stress chemistry that accompanied the original experience. The memory gets consolidated. You remember what happened. But the emotional charge gets gradually stripped away. The sting fades. The factual content persists. And this is where something really remarkable starts to emerge about how all these different levels of explanation fit together. Because you can tell the story of REM sleep at the level of brain regions: amygdala active, prefrontal cortex quiet, visual cortex generating imagery. You can tell it at the level of neurotransmitters: acetylcholine high, norepinephrine, and serotonin low. You can tell it at the level of synaptic plasticity: BDNF, mTOR, protein synthesis, selective consolidation. And you can tell it at the level of lived experience: dreams, emotional resolution, waking up with a fresh perspective on yesterday's problems. These aren't competing explanations. They're the same phenomenon described at different scales, like looking at a cathedral from across the city, then from the street, then examining the mortar between individual stones. Each view reveals something the others can't, and none of them alone gives you the complete picture. You need all of them, layered together, to really understand what's happening when your eyes start darting beneath closed lids, and your body goes still, and your brain lights up like a city at night. And here's what's beautiful about arriving at that understanding. It took us more than half a century to get here. From Oserinsky noticing those eye movements in 1953 to the molecular work of the 2010s, each decade added a new layer of resolution to the picture. And the picture is still being refined, still being debated, still surprising us. But one of the most fascinating things happening right now in sleep science is how researchers are taking all of these layers: the brain regions, the chemistry, the synaptic mechanics, the subjective experience, and using them to understand what goes wrong when REM sleep breaks. Because REM sleep does break. It breaks in specific, revealing ways, and understanding those breakdowns has become one of the most productive avenues in modern neuroscience and psychiatry. Let's start with something we touched on earlier: PTSD. Post-traumatic stress disorder is, in many ways, a disorder of failed emotional processing, and one of its hallmark features is disrupted REM sleep. People with PTSD often have fragmented REM periods. They wake up frequently during the night, particularly during or just after REM episodes, and their dreams, this is crucial, their dreams tend to be exact or near exact replays of the traumatic event, which is actually unusual. Normal dreaming, even when it incorporates elements of real experience, tends to be transformative. It recombines things. It takes a stressful meeting at work and turns it into a scene where you're giving a presentation in a grocery store, or something equally bizarre. That recombination, that creative remixing, appears to be part of how the brain processes and integrates emotional material. But in PTSD, that process seems to stall. The dream replays the trauma faithfully. The person wakes in terror, and the emotional charge of the memory doesn't diminish over time the way it normally would. Now think about what we know about the neurochemistry of REM sleep. In healthy REM, norepinephrine is essentially shut off. That's one of the defining features of the state. But there's evidence, and this is work that Murray Raskind at the VA Puget Sound was instrumental in developing. that in people with PTSD, norepinephrine levels remain elevated even during REM sleep. The locus coeruleus, that little brainstem structure that Jouvet and others identified as a key player in the REM switching mechanism, doesn't fully quiet down. So the brain is trying to do its emotional reprocessing. The amygdala is active, the memories are being replayed, but the neurochemical environment is wrong. The stress chemistry is still present. Instead of replaying the memory in a safe, norepinephrine-free context where the emotional edge can be gradually filed down, the brain is essentially re-traumatizing itself. Each replay reinforces the emotional charge rather than diminishing it. The person wakes up feeling not better, but worse. And the cycle continues, night after night. This is where prazosin comes back into the story. Raskind, working with combat veterans in the late 1990s and early 2000s, tried using prazosin, a drug originally developed for high blood pressure that works by blocking a specific type of norepinephrine receptor called the alpha-1 adrenergic receptor. The idea was elegant in its simplicity. If the problem is too much norepinephrine activity during REM sleep, what if you could pharmacologically recreate the low norepinephrine environment that healthy REM requires? And it worked. Veterans taking prazosin reported dramatic reductions in trauma nightmares. Their sleep improved. Their daytime PTSD symptoms got better. It was one of those moments where the basic science, all that painstaking work on neurotransmitter switching and REM physiology, directly informed a clinical intervention that changed people's lives. Now, the story got more complicated later. A large VA-funded clinical trial in 2018 didn't show the same robust effects, and there's been a lot of debate about why: differences in patient populations, dosing, the complexity of PTSD itself. But the underlying logic, the connection between REM neurochemistry and emotional processing, remains one of the most compelling frameworks in sleep medicine. And it illustrates something important about how science actually works. The path from basic research to clinical application is rarely straight. It's full of detours and disappointments and partial successes. But the fundamental insight that REM sleep provides a specific neurochemical environment for emotional memory processing, and that disrupting that environment has consequences. That insight has held up remarkably well. And PTSD isn't the only condition where REM sleep tells us something important. Let's talk about depression for a moment, because the relationship between REM sleep and depression is one of the strangest and most counterintuitive stories in all of sleep science. People with major depression tend to have altered REM sleep architecture. Specifically, they often show what's called shortened REM latency, meaning they enter their first REM period much sooner after falling asleep than healthy individuals do. Instead of the typical 90 minutes or so, they might hit REM in 30 or 40 minutes. And their early REM periods tend to be longer and more intense than normal. It's as if the brain is hungry for REM, rushing toward it, spending more time there early in the night. Here's where it gets really strange. One of the oldest and most reliable findings in biological psychiatry is that depriving a depressed person of sleep, particularly of REM sleep, can produce a rapid, dramatic improvement in mood. This was first reported systematically in the 1970s by researchers in Germany, and it's been replicated many times since. You take someone who's been profoundly depressed, you keep them awake all night, or selectively wake them every time they enter REM sleep, and by morning, a significant percentage of them feel substantially better. Not all of them, but enough that it's a real and reproducible phenomenon. Think about how weird that is. We've spent all this time talking about how REM sleep is essential for emotional processing, for stripping away the negative emotional charge from difficult memories, for maintaining psychological equilibrium. And yet, here's a situation where removing REM sleep actually improves someone's emotional state. What's going on? Well, the honest answer is that we don't fully understand it yet. But there are some intriguing ideas. One possibility is that in depression, the REM sleep system is actually malfunctioning. It's not doing its normal emotional processing work. It's doing something else. Something that reinforces depressive rumination rather than resolving it. Some researchers have suggested that the excessive early-onset REM sleep in depression might involve a kind of hyperactive emotional reprocessing that strengthens negative emotional associations rather than weakening them. If that's the case, then interrupting that process, breaking the cycle, could provide temporary relief. It's a bit like the difference between productively thinking through a problem and obsessively ruminating on it. Same brain regions might be involved, but the outcome is completely different depending on the mode. And here's an interesting connection that took decades to notice. Many of the most effective antidepressant medications, SSRIs, SNRIs, MAO inhibitors, tricyclics, they all suppress REM sleep. Dramatically, in many cases. When someone starts taking an SSRI like fluoxetine, their REM sleep drops significantly, sometimes by 50% or more. For years, this was treated as a side effect, something irrelevant to the therapeutic mechanism. But some researchers, including work by Vogel in the 1970s and 1980s, argued that the REM suppression wasn't a side effect at all. It might be part of how these drugs actually work. That's a provocative claim, and it's still debated. But the correlation is striking. Almost every class of effective antidepressant suppresses REM sleep. The exceptions are few. Now, you don't want to take this too far. Correlation isn't causation, and antidepressants do many other things besides suppressing REM. They alter serotonin and norepinephrine signaling in ways that affect waking brain function too. But the pattern is suggestive enough that it keeps researchers thinking about the relationship between REM sleep and mood regulation in more nuanced ways than a simple REM sleep is good for your emotions story would suggest. And this is actually one of the most important things to understand about REM sleep as we know it today. It's not simply good or bad, helpful or harmful. It's a biological process that can function well or poorly, and the difference between those outcomes depends on the precise neurochemical conditions, the prior experiences being processed, the integrity of the underlying circuits, and probably factors we haven't identified yet. It's like saying exercise is good for you. That's broadly true, but the specifics matter enormously. The type, the intensity, the timing, the individual's health status. REM sleep is the same way. When it's working properly, it appears to do remarkable things for memory consolidation, emotional regulation, and creative problem solving. When it's not working properly, when the chemistry is off, when the timing is disrupted, when the content gets stuck in repetitive loops, it can contribute to pathology rather than healing. This more nuanced view has been one of the major shifts in sleep science over the past decade or so. The field has moved away from simple, more sleep is always better messaging, though getting enough sleep is genuinely important, don't get me wrong, toward a more sophisticated understanding of sleep quality, sleep architecture, and the specific functions of different sleep stages. And REM sleep, because of its unique neurochemistry and its deep connections to emotional and cognitive function, has been at the center of that shift. Which brings us to another frontier that's been developing rapidly: the relationship between REM sleep and neurodegenerative disease. And this story starts with something we mentioned much earlier, almost in passing: REM sleep behavior disorder, the condition where the normal muscle paralysis of REM sleep fails and people physically act out their dreams. Remember Schenk and Mahowald documenting those cases in Minnesota back in the 1980s? Well, in the decades since that initial description, something deeply unsettling has emerged from the long-term follow-up of those patients. It turns out that REM sleep behavior disorder is one of the strongest known predictors of future Parkinson's disease and a related condition called dementia with Lewy bodies. The numbers are sobering. Studies following patients for 10, 15, 20 years have found that somewhere around 80% or more of people diagnosed with isolated REM sleep behavior disorder eventually go on to develop one of these neurodegenerative conditions. And the sleep disorder often appears years, sometimes more than a decade, before any of the classic motor symptoms of Parkinson's: the tremor, the rigidity, the shuffling gait. Think about what that means. The brainstem circuits that control rematonia, the ones that keep your muscles paralyzed while you dream, appear to be among the first neural structures damaged by the disease process that eventually becomes Parkinson's. The alpha-synuclein protein aggregates that characterize Lewy body pathology seem to spread through the brain in a somewhat predictable pattern, and the pontine structures involved in REM sleep regulation are hit early. So the breakdown of rematonia isn't just a sleep problem. It's a window into a neurodegenerative process that's already underway, years before it becomes clinically obvious through other symptoms. This has enormous implications for early detection and potentially for early intervention if disease-modifying therapies for Parkinson's can be developed. Several major research consortia around the world are now following large cohorts of people with REM sleep behavior disorder, tracking them with neuroimaging, biochemical markers, and clinical assessments, trying to understand exactly when and how the transition to full neurodegenerative disease occurs. It's turned what was once considered a curious sleep oddity into one of the most important biomarkers in neurology. And there's something poignant about this connection, if you think about it. The very mechanism that protects you during your most vivid, emotionally charged mental experiences, the paralysis that keeps your dreaming mind safely contained within your still body, turns out to be a canary in the coal mine for one of the most feared diseases of aging. The brainstem nuclei that Juve mapped in his cats in the 1960s, the ones that create what he called paradoxical sleep, are telling us something about the vulnerability of the aging brain that we're only now beginning to fully understand. And that understanding is expanding in directions that would have seemed like science fiction even 20 years ago. Because here's the thing. Once researchers recognized that REM sleep behavior disorder could serve as this remarkably early warning sign, they started asking a much bigger question. Not just, can we predict who will develop Parkinson's? But, can we intervene during that window, during those years when the disease is silently spreading but hasn't yet caused the damage that leads to tremor and rigidity? And that question connects to one of the most ambitious frontiers in modern neuroscience: the idea that sleep itself might not just reflect brain health, but actively shape it, for better or worse, across the entire lifespan. Let's sit with that idea for a moment, because it pulls together so many threads we've been following. We talked earlier about how REM sleep helps consolidate memories, how it strips the emotional charge from difficult experiences, how it bathes developing neural circuits and the kind of activity they need to wire up properly. All of those functions depend on REM working the way it's supposed to. The right neurochemical environment, the right patterns of activation, the right timing within the overall architecture of sleep. So what happens when REM starts going wrong? Not dramatically, not in the way that brings someone to a sleep clinic, but subtly, gradually, over years and decades. This is where the story of REM sleep connects to one of the hottest areas in aging research. There's been a growing recognition, really gaining momentum since the early 2000s, that sleep quality doesn't just decline as a consequence of aging. It may actually accelerate the aging process itself, particularly in the brain. And REM sleep is right at the center of this conversation. You've probably heard the broad outlines. Matthew Walker and others have been vocal about the links between poor sleep and Alzheimer's disease. But let's get specific about the REM piece, because it's more nuanced than the popular coverage sometimes suggests. The relationship between sleep and Alzheimer's involves multiple sleep stages, but REM has its own particular story. A study published in 2017 by Matthew Pace and colleagues, using data from the Framingham Heart Study, one of the longest-running epidemiological studies in the world, tracking residents of Framingham, Massachusetts since 1948, found that people who got less REM sleep had a higher risk of developing dementia over the following years. And here's what made the finding striking. It was specific to REM. The association held even after controlling for other sleep stages. Less time in REM, higher dementia risk. Not less time in deep, slow-wave sleep. Not more time awake during the night. Specifically, less REM. Now you have to be careful with findings like this. Epidemiological associations don't tell you which way the causation runs. It could be that reduced REM sleep contributes to dementia. It could be that the earliest subclinical stages of dementia are already disrupting REM circuits before anyone notices cognitive decline. It could be both. A vicious cycle where each makes the other worse. And honestly, the evidence increasingly points toward that bidirectional relationship, which makes it both more interesting and harder to untangle. But think about what we know about what REM sleep does. We talked about the overnight therapy hypothesis, the idea that REM provides this neurochemically unique environment where emotional memories can be reprocessed without the stress chemicals that accompanied the original experience. We talked about synaptic homeostasis, about how sleep helps recalibrate the connections between neurons. We talked about the role of REM in integrating new information with existing knowledge, finding hidden patterns, making creative connections. All of these processes matter for maintaining a healthy brain as you age. If REM is gradually eroding, less of it each night, or the REM you do get is more fragmented, less neurochemically clean, then you're losing a maintenance process that the brain depends on. And here's something that doesn't get discussed enough in the popular accounts. The decline in sleep quality with aging isn't uniform across all stages. Slow-wave sleep, the deep, restorative non-REM sleep, drops off fairly dramatically starting in middle age. By the time you're in your 70s, you might be getting a fraction of the deep sleep you got in your 20s. REM sleep, interestingly, is somewhat more preserved. It does decline, but not as steeply. However, and this is the key point, when REM does decline, or when its quality degrades, the consequences may be particularly significant precisely because it's doing things that no other sleep stage does. That emotional processing, that specific pattern of acetylcholine-rich, norepinephrine-poor brain activity, that vivid internal simulation, There's no backup system for those functions. If REM goes, those processes go with it. So researchers started asking, could protecting or restoring REM sleep slow cognitive decline? Could it change the trajectory of neurodegenerative disease? And this is where the story gets both exciting and frustrating, because the honest answer is we don't fully know yet. But there are some fascinating clues. One line of evidence comes from the relationship between REM sleep and the glymphatic system. And if you haven't heard of this, it's one of the most remarkable discoveries in neuroscience in the past decade. In 2012, Maiken Nedergaard and her team at the University of Rochester published work showing that the brain has its own waste clearance system, distinct from the lymphatic system that serves the rest of the body. They called it the glymphatic system. The G is for glia, the non-neuronal brain cells that help manage this process. During sleep, the spaces between brain cells expand, and cerebrospinal fluid flows through in pulsing waves, flushing out metabolic waste products. Among the waste products cleared by this system is beta amyloid, the protein that accumulates in the plaques, characteristic of Alzheimer's disease. Now, most of the initial work on glymphatic clearance focused on non-REM sleep, particularly deep slow-wave sleep, which seemed to be the primary state during which this flushing process operated. But more recent work has complicated the picture. Studies have suggested that the transitions between sleep stages and the overall integrity of sleep architecture, including REM, matter for efficient waste clearance. If your sleep is fragmented, if you're not cycling smoothly through the stages, the glymphatic system doesn't work as well. And REM sleep, which occurs in longer bouts toward the end of the night, may play a role in the later phases of this clearance process. So when people cut their sleep short, setting an alarm that wakes them during what would have been their longest REM period of the night, they may be sacrificing not just dream time, but waste clearance time. Think about what that means in practical terms. Every time you set your alarm for 6 hours instead of 7.5, you're probably cutting into your richest REM period. And over years and decades, that cumulative loss of REM and the brain maintenance it supports might matter more than anyone realized until recently. But let's come back to the clinical side, because there's another connection between REM sleep and modern medicine that's worth exploring. And it involves something millions of people encounter every day: medication. An enormous number of commonly prescribed drugs affect REM sleep, and in many cases, neither the doctors prescribing them nor the patients taking them are fully aware of this. Antidepressants are the most obvious example, and we touched on this earlier when we talked about the paradox of REM suppression improving depression. Most SSRIs, selective serotonin reuptake inhibitors, the most commonly prescribed class of antidepressants, significantly reduce REM sleep. Some of them cut REM time by 30, 40, even 50%. SNRs do the same. Older tricyclic antidepressants do the same. MAO inhibitors can virtually eliminate REM sleep entirely. And here's what's remarkable. For most patients, this doesn't seem to cause obvious problems in the short term. People take these medications for years, with dramatically reduced REM sleep, and they function. They don't develop the hallucinations and cognitive fragmentation you see in total REM deprivation experiments. Why not? This is genuinely one of the open questions in sleep science. One possibility is that the brain compensates, that the remaining REM sleep becomes more intense, more efficient, packing more of the essential processing into less time. There's some evidence for this. Another possibility is that the neurochemical changes caused by the antidepressants themselves partially substitute for some of what REM normally does. that by altering serotonin and norepinephrine levels around the clock, these drugs are, in a sense, doing chemically what REM does architecturally. A third possibility, and this is the one that keeps some researchers up at night, no pun intended, is that there are long-term consequences of chronic REM suppression that we simply haven't tracked carefully enough. Maybe the effects are subtle, accumulating over years, showing up as slightly faster cognitive decline, slightly less emotional resilience, slightly impaired creative problem-solving in ways that are real but hard to pin on any single cause. This isn't meant to alarm anyone who's taking antidepressants, by the way. These medications help millions of people, and the benefits of treating depression, which is itself devastating to brain health, almost certainly outweigh the costs of reduced REM in most cases. But it's a reminder of how much we still don't understand about what happens when you chronically alter one of the brain's fundamental operating modes. And it's not just antidepressants. Beta blockers, commonly prescribed for high blood pressure and heart conditions, can suppress REM sleep. So can many anti-anxiety medications. Alcohol, which so many people use as a sleep aid, is a potent REM suppressant, particularly in the first half of the night. Cannabis, increasingly legal and widely used, also appears to reduce REM sleep, which is why heavy users often report an explosion of vivid dreams when they stop. The classic REM rebound effect, the brain catching up on what it's been missing. So here we are, in an era where we understand more about REM sleep than at any point in human history. And yet we're also living in ways that may be systematically undermining it. Through shortened sleep, through medications, through substances, through the blue light from screens that shifts our circadian rhythms and compresses our morning REM periods. There's an irony there that's hard to ignore. But the story isn't all cautionary. Because the same depth of understanding that reveals these risks also opens up possibilities that earlier generations of sleep researchers couldn't have imagined. And some of the most exciting work happening right now involves not just protecting REM sleep, but actually enhancing it using techniques that range from remarkably low-tech to genuinely futuristic. One approach that's gained traction involves targeted memory reactivation during sleep. The basic idea is beautifully simple. You expose someone to a specific sensory cue, a particular smell, a particular sound, while they're learning something new during the day. Then, while they're asleep, you present that same cue again, timed to coincide with specific sleep stages. The cue seems to reactivate the associated memory, boosting the consolidation process. Most of this work has focused on slow-wave sleep, but researchers are beginning to explore whether similar techniques could enhance REM-dependent processing, emotional memory integration, creative problem-solving, the kinds of things REM seems uniquely suited for. And then there's the more ambitious frontier, closed-loop stimulation. This involves monitoring someone's brain activity in real time during sleep and delivering precisely timed stimulation, electrical, auditory, or otherwise, to enhance the natural rhythms of specific sleep stages. For slow-wave sleep, this has already shown promising results. Gentle auditory tones, played in sync with the brain's own slow oscillations, can deepen and extend those oscillations. The question now is whether similar approaches could work for REM, whether you could detect the onset of REM, monitor its quality, and subtly support or extend it when it starts to fragment. The technical challenges are significant because REM's brain activity patterns are more complex and variable than the steady rhythms of deep sleep. But the potential is enormous, because if you could gently coax someone's REM sleep to be longer, more stable, more architecturally intact, you'd potentially be improving everything from their emotional resilience to their creative capacity to their long-term neurological health. And the early attempts are genuinely fascinating. Some groups are experimenting with transcranial direct current stimulation, very mild electrical currents applied to the scalp, timed to the onset of REM periods. Others are looking at whether specific auditory patterns, not the slow rhythmic tones that work for deep sleep, but something more complex, maybe even something melodic, could entrain or stabilize REM activity without waking the person up. That's the tightrope, right? REM is already so close to waking consciousness that any intervention risks tipping someone right over the edge into actual wakefulness. You're trying to support a state that's inherently fragile, that exists in this narrow corridor between deep sleep and alertness, and any nudge that's too strong just collapses the whole thing. There's a group at the University of Freiburg that published some intriguing work around 2020 looking at transcranial alternating current stimulation during REM sleep. They were trying to influence the gamma frequency activity that characterizes lucid dreaming. That's a related but slightly different question, whether you can induce awareness within the dream state. And they found some evidence that stimulation at around 25 to 40 Hz during REM could increase the likelihood of lucid dreaming. Now, lucid dreaming isn't the same thing as enhancing REM's restorative functions, but it demonstrates something important: that external stimulation can modulate what's happening inside REM without necessarily destroying it. That's a proof of concept. It tells us the door is open, even if we haven't figured out exactly how to walk through it yet. And here's where things get really interesting from a practical standpoint. Think about the population that might benefit most from REM enhancement. It's not healthy young adults who already get plenty of REM. It's older adults whose REM is naturally declining. It's people with PTSD whose REM is fragmenting under the weight of persistent norepinephrine. It's people on medications that suppress REM. The millions of people taking SSRIs or SNRIs who might be getting adequate total sleep, but whose REM architecture is quietly eroding. If you could develop a reliable, non-invasive way to support REM in those populations, the clinical implications would be staggering. But let's be honest about where the field actually stands right now, because there's a gap between the excitement of the research and what's available in practice. As of the early 2020s, there is no clinically validated REM enhancement technology that you can buy or that your doctor can prescribe. The consumer sleep technology market is full of devices that claim to improve sleep quality: headbands, rings, apps, mattress pads. And some of them do a reasonable job of tracking sleep stages, but none of them have demonstrated the ability to selectively enhance REM sleep in a way that's been rigorously validated. That doesn't mean they're useless. Better sleep tracking can lead to better sleep habits, and better sleep habits can lead to more REM. But the direct enhancement piece is still in the laboratory. And honestly, the most effective REM enhancement strategy available to most people right now is embarrassingly simple. It's just protecting the conditions that allow REM to happen naturally. And that means understanding something about when REM occurs in the night, which brings us back to something we touched on much earlier: the architecture of sleep across the night. Remember how sleep cycles work. You go through roughly 90-minute cycles, each containing some mix of light sleep, deep sleep, and REM. But the proportion shifts dramatically across the night. Your early cycles are dominated by deep, slow-wave sleep. Your brain prioritizes that first: the metabolic restoration, the synaptic downscaling that Tononi and Sorelli described, the heavy-duty housekeeping. REM shows up in those early cycles, but it's brief, maybe 10 minutes. As the night goes on, the balance shifts. By the time you're in your 5th or 6th cycle, typically in the last 2 or 3 hours of an 8-hour sleep period, REM dominates. Those late morning REM periods can last 30, 40, even 50 minutes. They're the longest, most vivid, most architecturally complex REM periods of the night. So here's what that means in practical terms. If you cut your sleep short, if you sleep 6 hours instead of 8, you're not losing a proportional amount of each sleep stage. You're disproportionately losing REM because you're cutting off exactly the part of the night where REM is most abundant. That alarm clock going off at 6 a.m. when your body wanted to sleep until 8 isn't just costing you 2 hours of sleep. It's potentially costing you your 2 richest REM periods, the ones where the most emotional processing happens, the ones where the most creative integration occurs, the ones where your brain is doing its most sophisticated overnight work. And this is one of those places where the science of sleep bumps up against the reality of modern life in a way that's genuinely uncomfortable, because most people in industrialized societies are chronically sleep restricted. Not dramatically, not pulling all-nighters regularly, but shaving an hour or 2 off the end of their sleep most nights. And that pattern, sustained over months and years, creates a selective REM deficit that accumulates quietly. You might feel tired, sure, but the specific consequences of REM loss, the emotional dysregulation, the impaired creativity, the reduced ability to integrate new experiences into your existing understanding of the world, those are subtle. They don't announce themselves the way total sleep deprivation does. They just slowly erode the quality of your waking life in ways that are hard to attribute to any single cause. Matthew Walker has talked about this extensively, and while some of his more dramatic claims have been debated, and it's worth noting that sleep science, like any field, has its share of contested findings and replication challenges, the basic observation is hard to argue with. Modern life systematically steals REM sleep, and we're only beginning to understand what that costs us. So where does all of this leave us after everything we've wandered through tonight? From Hansberger's quiet laboratory in Jena, through Oserinsky's lucky observation of his sleeping son, through Jouvet's cats and Hobson's brainstem maps, and Stickgold's Tetris dreams, and Tononi's synaptic homeostasis, and Nedergaard's glymphatic rivers. After all of that, what do we actually know about REM sleep? Well, here's what I think is fair to say. We know that REM sleep is a distinct and ancient brain state, one that evolved hundreds of millions of years ago and has been conserved across an extraordinary range of species. We know it's characterized by a unique neurochemical environment, rich in acetylcholine, stripped of norepinephrine and serotonin, that creates conditions found in no other state of consciousness. We know it involves widespread brain activation, particularly in emotional and memory-related circuits, combined with almost total paralysis of the voluntary muscles. We know it's when most vivid dreaming occurs, though dreaming isn't exclusive to it. We know that REM sleep plays important roles in emotional processing, that it seems to help strip the emotional charge from difficult experiences while preserving the informational content. We know it contributes to memory consolidation, particularly for procedural skills, emotional memories, and the kind of creative, associative thinking that connects disparate ideas. We know it's involved in brain development, that the developing brain floods itself with REM in a way that suggests it's essential for building and refining neural circuits. And we know that when REM goes wrong, when it fragments, when the atonia fails, when it's suppressed or distorted, the consequences can range from nightmares and emotional instability to potential long-term neurological vulnerability. But here's what I think is equally important to acknowledge. We don't fully understand REM sleep. Not yet. We don't have a single unified theory that explains why this state exists, what it's optimizing for, or how all its various functions fit together. The emotional processing hypothesis, the memory consolidation hypothesis, the synaptic homeostasis hypothesis, the developmental hypothesis, the threat simulation hypothesis. They're not necessarily competing. They might all be partially right, capturing different facets of something too complex for any single framework. REM sleep might be doing many things simultaneously. The way your cardiovascular system simultaneously delivers oxygen, removes waste, distributes hormones, and regulates temperature. Asking, what is the function of REM sleep, might be like asking, what is the function of blood flow? The answer is, it depends on where you look and what you measure. And that's okay. That's what makes this field so alive right now. We're in a period where the tools, optogenetics, high-density EEG, functional neuroimaging, machine learning applied to sleep data, molecular techniques that can tag and track individual synapses, are finally catching up to the complexity of the questions. The next 20 years of REM research will almost certainly overturn some of what we think we know now and reveal dimensions we haven't even imagined yet. But tonight, lying here, you don't need to solve any of those questions. You just need to let your brain do what it already knows how to do. Because that's maybe the most remarkable thing about everything we've talked about. All of this, the neurochemical switching, the muscle atonia, the memory replay, the emotional recalibration, the synaptic tuning, the glymphatic clearing. All of it happens without your conscious participation. You don't have to direct it. You don't have to understand it for it to work. Your brain has been doing this every single night of your life, from before you were born. Running these ancient programs with a precision and sophistication that our best neuroscience can describe, but not yet fully explain. Right now, as you lie here, your brain is already preparing for its nightly work. The adenosine that's been building up all day is pressing on your arousal circuits, nudging you towards sleep. Your body temperature is dropping, your melatonin is rising, and somewhere in your brainstem, the flip-flop switch that Saper described is tilting toward its sleep-promoting side. Soon, maybe very soon, you'll cross that threshold, and the first slow waves will begin to roll across your cortex like gentle swells on a calm ocean. Your brain will begin its deep housekeeping, the synaptic downscaling, the metabolic restoration, the clearing of the day's accumulated debris. And then, after an hour or so, something will shift. The pontine neurons will begin to fire. Acetylcholine will flood your forebrain. Your eyes will begin to move beneath your closed lids, and your muscles will go slack, and your brain will light up with activity that looks, from the outside, almost indistinguishable from waking. But you won't be awake. You'll be somewhere else entirely, in that strange, vivid, emotionally saturated world that REM creates. A world where the rules of physics are optional, and the boundaries between memories are porous, and the feelings are overwhelming and real even when the scenarios are impossible. And your brain will be working, quietly, efficiently, brilliantly, replaying the day's experiences, stripping away what doesn't matter, strengthening what does, softening the sharp edges of difficult emotions, finding connections between ideas that your waking mind was too focused, too linear, too norepinephrine saturated to see, building and pruning and integrating, all while you dream about flying or showing up late to an exam you didn't study for, or having a conversation with someone you haven't seen in years. You don't need to do anything. You don't need to remember any of this. The science is fascinating, but the sleep is what matters, and the sleep will come on its own. It always does. Your brain has been practicing this for your entire life, and it's very, very good at it. So let it work. Let the thoughts soften. Let the details blur. Let Hans Berger and his string galvanometer, and Oserinsky's sleeping sun and Jouvet's cats and all the rest of it fade into the background, like the last light of a long day dimming at the horizon. They'll still be there tomorrow if you want them, but right now, there's nothing to figure out, nothing to solve, nothing to remember. Just the quiet, ancient, extraordinary machinery of your sleeping brain, doing what it was built to do.